Good morning. So I'm Jan Apkowitz. I'm head of the Division of Hematology, and it's my pleasure today to introduce the Clem Finch Visiting Professor for 2014 and to say a few words about Clem, who I know is known to several of you in the audience. So Clem was one of the initial founders of the medical school. He started the Division of Hematology in 1949 and remarkably served as its chair for 32 years. Which many of his predecessors like me think is quite a remarkable experience that he did. In the earlier years, he was actually key to the establishment of the University of Washington Medical Center, and he wrote a history of the UW Medical Center that's actually quite a novel one if you want to learn about our beginnings. And he made many contributions to hematology. He's really the grandfather of hematology, thought of as the Iron Man of hematology because of his focus on iron. And he defined iron kinetics and physiology. He studied transferrin biology at a very early time, but perhaps most importantly, because of his really brilliant physiologic studies, he defined physiologically mediators like ferroportin and hepcidin that many years later were molecularly defined. But their physiology and how they worked were absolutely identical to the molecules that he predicted had to exist. He also described red cell development and the physiology of a number of anemias. He showed that hemochromatosis was due to the hyperabsorption of iron in the GI tract, which was quite counterintuitive since it was thought to be a disease of liver storage at the time, and that it could be treated with phlebotomy. He defined many better ways to store blood, so he was very instrumental in blood banking practices. And he also was very active, extraordinarily active, and received several medals from the World Health Organization for his work on nutritional anemia globally. He's really one of the beginners of global medicine. Because he championed iron supplementation of food, which had a major impact in the developing world. So my memories of Clem, I was actually his last fellow, his last clinical fellow. And so that dates him. He stayed around for a bit afterwards. He was known to many people subsequent to me, but I feel very privileged to have worked with him as a recruit for a clinical fellowship program and then to have him be my attending. So he was very tall in the world of academic medicine and also just in general. Very smart, very creative, a creative scientist and just a creative person, an excellent observer and listener. And it could put together physiology because he was able to observe it both in patients and in animal and cell models, and very eclectic in his interests. And so it's a pleasure today to introduce Neal Young, who is this year's Clem Finch Visiting Professor, because in many ways he's like Clem, except he's not very tall. So Neal, he's not very short either. He's very smart. He's very creative. He's an excellent observer of physiology and of patients and integrates patient care with spectacular thinking of basic science. And eclectic in his interests. I don't know if he'll share that, but I do know that because I've known Neal for quite a long time. So Neal went to medical school at Harvard Medical School. He was a house staff at MGH, and then went to Hopkins for his Harvard Hopkins MGH. Yeah, Harvard Hopkins for initial time, and MGH for residency, and then to St. Louis for fellowship. All the places that are basically on our roadmap of hematology programs, besides ours in San Francisco, ones in the world, and then ended up at the NIH Hematology branch, where he stayed for his entire academic career. And so he is currently chief of the hematology branch. He's contributed big time to a number of NIH activities that are broader than NHLBI. He is very well known in hematology circles and has had many national presentations at the ASCI. He's a counselor. He's been an editor of Blood. He's been a major member of the Plastic Anemia Foundation, and contributed on many, many levels to our understanding of marrow failure, of telomeres and telomerase, which is going to speak about today, cancer development, clonal evolution, aplastic anemia, and spanning really the whole viruses in hematology, and spanning the entire premise from basic science to patient application. And so it's a great pleasure to introduce Neal today.
It's always daunting to be compared to a great, and thank you, that's extremely complimentary, that's the truth. I actually met Clem Finch towards the end of his career when I was looking at fellowships and thought of coming to the University of Washington. I was interviewing here. At that time, there was iron and there was a bone marrow transplant, so I just wanted something that exists between a rock and a hard place. Those of you who do transplant, and that's the case. I know it does require some booze, so I ended up not here. But he was wonderful and very one of those open, sweet, intelligent people, just exactly as described. So very great gratitude for the invitation here. It is an honor. So let me put up my own talk. This is really tough, this Clem Finch visiting fellowship. I really hope that I have a chance to go sometime to the bathroom today. It's a very full schedule, and I was instructed that I had grand rounds today and then would be at the Hutch tomorrow. Can't be on the same subject, and I was told I was going to be on the Seattle Blood Club. That can't be on the same subject, and then I talked to the fellows, and that can't be on the same subject. So at some point, you run out of subjects that you can speak about knowledgeably. And so I have to do this kind of dance of putting some slides in from tomorrow. That'll be in here today. So if you happen to come to the talk tomorrow, just forget that, and vice versa. These are all interconnected issues, and I'll tell you a little bit about aplastic anemia just as an entry. But my main goals are, as they're outlined here, is to really describe to you a new group of diseases, the telomeropathies, which are genetically based, and also, and this is separate, the role of telomere loss that occurs physiologically, we think independently of the genetics that contribute to cancer and to organ failure. So I start with aplastic anemia because that's in fact what I've been studying for all these years. And it is bench to bedside, bedside to bench. A lot of my career has been from the bedside to the bench, and this is a sort of a nice early version. This is a nice picture of what Paul Ehrlich saw 100 years ago with a much more primitive microscope. It's a confocal image of a human bone marrow that is suffering from aplastic anemia. Those are the CD34 endothelial cells that are lighting up. There are no hematopoietic progenitor and stem cells that are detectable in the marrow, which should be full of nice blue cells. Instead, it's replaced by fat in this reconstruction. Really hard disease to study because we don't have any tissue and because the patients die quickly. And in fact, when I started to study aplastic anemia back at the sort of latter period of Professor Finch's career, everybody died. It was really a dismal disease to take care of, and all those people mainly were young. So older adolescents and young adults who would be struck out of the blue with this disease, which was fatal within a year. Now, I'll spend a lot of time tomorrow and very little time today saying that this is really one of the great success stories in hematology and in modern medicine. The observation initially in Europe and then repeated here that patients could respond to immunological therapy. No reason to think this was an immunological disease because the marrow was gone, and there's nothing to suggest that this was anything to do with the immune system, but these patients did respond to relatively gentle immunosuppressive therapy, as shown in this example of one of our patients, who actually is from the state of Washington, who had post-hepatitis aplastic anemia. You see this lovely improvement in his blood counts, which is permanent over the course of multiple decades with a very brief period of anti-thymus cycloglobulin followed by cyclosporin. And we've now reversed the survival curves for patients with this disease, most of whom respond to immunosuppression, and when they do respond, they do marvelously well, with greater than 90% long-term survival in our most recent cohorts. That's the good story. I'll tell that tomorrow. Now, the emphasis, because of this dramatic response to immunological therapy, has been on defining T-cell clones, cytotoxic lymphocytes, cytokines, what causes the marrow to be destroyed. And there's been the assumption that the hematopoietic cells are more or less passive and there's not much to be done about them because, in fact, they're gone. So we think about this disease, at least historically, in very straightforward ways. There's some sort of inciting event. This balances the immune system, leading to a destructive process in the bone marrow. You lose your stem cells and can be salvaged either by a transplant, replace both the hematopoietic and the immune systems, or by immunosuppressive therapy. There's a problem with immunosuppressive therapy, which has been a difficult one, which is that a minority, but a substantial minority of patients over time evolve from what seems to be this immune-mediated disease responsive to appropriate therapies to MDS, myelodysplastic syndrome, and acute leukemia. That's been a mystery as well as being a clinical disaster for the patient and to some extent the physician. But we've also begun to appreciate that not only is aplastic anemia related to other T-cell mediated organ-specific diseases. I've shown those on the left, multiple sclerosis, inflammatory bowel disease, all have pathophysiologic features that are similar and can respond to similar sorts of treatment. But we've also begun to appreciate that there's also a relationship between aplastic anemia and genetic syndromes in which there are very clearly described genes, the telomere repair genes that I'm going to discuss in a few moments, that inform the pathophysiology of this disease and also its treatment. Now, the link between telomeres and aplastic anemia is to some extent accidental, and it comes from two sources. One is that telomeres, which I'm going to define for you in a moment, were noted to be short, or telomere content was noted to be low, in some patients with acquired aplastic anemia. Now, why were these experiments done? They were done because the assays happened to be available. So both the group at St. George's at that time in London and then Peter Lansdorp and the group at NIH showed that we had short telomeres in a substantial minority with aplastic anemia. Complementary to that were investigations of genetic linkage in children with a very discrete form of congenital bone marrow failure called DYSKERATOSIS congenita. This is one of those diseases that adult hematologists remember only vaguely. Now it's a big deal, but in the old days it was just one of those funny, rare syndromes in which you were taught to look at the patient's fingernails. They look like they've been chewed to death, and in fact the first cases of DYSKERATOSIS congenita are in the dermatologic literature, because the nails were the findings that often brought children to medical attention. I've shown you the classic triad at the bottom left, and on the right, these measurements, these early measurements of short telomeres and acquired aplastic anemia. So Inderjeet Dokal, working in London with very large pedigrees, was just through very straightforward genetic linkage analysis surprised to find a gene which was later called DKC1, which was related to the telomerase, to the telomere repair complex. And sure enough, went back after we had shown that telomeres were short in aplastic anemia and found that children with DYSKERATOSIS congenita had very short telomeres. So it's from these two directions that the linkage between bone marrow failure and short telomeres occurred. Not really clear in the adults exactly what was going on, but in the children, seemingly this inherited inability to repair telomeres. Now, this is a simple but important slide to kind of guide you through what I'm going to say, because it's easy, even though I think the biology is really simple, it's easy to get confused. So I've told you that there's a link with aplastic anemia, and I'm going to demonstrate that patients who have defects in their telomere repair genes are predisposed, not destined, but predisposed to develop bone marrow failure. I'm also going to suggest a link between telomere attrition that occurs as a result of this genetic problem and the development of cancer. But this important second point is that telomere loss can be accelerated because of a genetic reason. The patient doesn't have adequate telomere repair, but telomeres can also be lost physiologically. In fact, I'm going to tell you that we're all losing our telomeres physiologically as we sit here for this hour, and that is independent of the genetics. There's this problem of the late development of cancer and aplastic anemia, as well as the bone marrow failure that characterizes it. William Damashek, sort of Finch's contemporary on the eastern side of the country, wrote extraordinarily prescient editorials in the early editions of Blood, linking aplastic anemia, other clonal diseases of the bone marrow, and leukemia. I and others have falteringly attempted to come up with some sort of physiologic link. And what I'm going to suggest also in this talk is not only is there this predisposition to bone marrow failure as a result of telomere loss, but a predisposition also to cancer, and that the problem of clonality that we see in hematology is in fact a fundamental biological problem with its answer in telomere biology. So where did this come from? Well, telomeres were actually suggested even before DNA was discovered by two Americans, actually, Herman Mueller and Barbara McClintock, working on chromosomes, before chromosomes were even understood to be made up of DNA. And McClintock recognized that there was something different about chromosome fragments that could be generated in maize, and Mueller with radiation damage to cells, and the natural ends, the term that McClintock used, of normal chromosomes. When DNA was discovered, as I'll show you in a moment, and the replication scheme was understood, it also was realized that there was a fundamental problem with DNA replication that had to do with the ends of the chromosomes. That was done both theoretically and also on experimental grounds, and all of these, with the exception of poor Alexei Alovnikov, all of these individuals went on to win Nobel Prizes for these very fundamental biological phenomena. So here's a picture of both McClintock and Mueller taken in the 1940s and the 1950s, doing their sort of one-person experiments and recognizing that normal chromosomes, natural chromosomes, are different from chromosome fragments that can be generated in tissue culture. This is an early paper of Alexei Olovnikov. He's Russian, so he published in Russian. Nobody noticed this paper. But he developed this theory of marginotomy after DNA replication was understood. The idea that there were complementary strands, that the DNA polymerase only ran along one strand and that fragments had to be generated to replicate the second strand. So, of course, nobody noticed this. Supposedly, in Olovnikov's autobiography, he says this idea of the problem of marginotomy, the problem with replication of the ends of the chromosomes, came to him on the Moscow Metro. This is a picture of the Moscow Metro, and the story is that he realized that the train's front arrived before the doors arrived. There was going to be this gap at the end of the train, at the end of the chromosome. And when he finally published this paper in English a couple of years later, I think these do look a little bit like trains on a track. And this is really still a paper, despite the fact that it's decades old, that's worth going back and reading, because Olovnikov really understood that this problem of marginotomy, the problem of replicating the ends of the chromosomes, would have implications for aging, for organ function, and for cancer. In the interval, Watson, working experimentally on phage bacteria, also realized that there were problems with the replication scheme that nature had endowed cells with linear chromosomes. And I'll just read for those in the back what Watson said in this solo author Nature paper. While 5' 3' oriented growth should proceed smoothly to the end of its template, DNA polymerase just churning out more synthetic nucleotides, I see no simple way for 3', 5' growth, the complementary strand, to reach the 3' end of its template. In other words, at the end of every mitosis, at the end of every replication cycle, there would be inevitable loss, this marginotomy problem of Olovnikov's, loss of the DNA at one of the termini. So how do we solve this? Well, this is the group that won the Nobel Prize a few years ago. Elizabeth Blackburn and her colleagues, especially Carol Ryder, recognized that there was a molecular engine as well as a specific structure at the end of the chromosome that led to preservation of this DNA material. So what is the telomere? The telomere is hundreds, even thousands of repeated hexanucleotide sequences. It's really not a lot more complicated than that. So this is in itself one mechanism by which naturally we avoid the problem of marginotomy. If there's material lost, it's junk. We don't have our beauty and our intelligence, our get into medical school genes, for example, are not at the ends of the chromosomes. They're much more central. So if there is fraying, think about the shoelace. If there's fraying at the end of the shoelace, it doesn't affect the core structure of the chromosome. The second component of the telomere is a group of proteins collectively termed shelterin, which coat this structure, forming a T-loop actually of these hexanucleotides, and signals to the cell that this is not damaged DNA or a fragment of a chromosome that needs to be removed or a DNA virus, or that it's material that is natural and should be left alone. So the shelterin proteins are part of the telomere structure. So that's a more or less passive mechanism. Nature has provided the chromosome with a specific structure that's distinctive and serves this. But in addition, the discovery of Blackburn and colleagues is that there's an active process of repair. And this is, again, very straightforward. The process is based on reverse transcription from an RNA template. That template is encoded by a gene called TERK. This is really a test of the speaker's enunciation to keep this straight because they really sound similar. TERK, T-E-R-C, is the gene that produces an RNA. And that RNA serves as the template for reverse transcription, absolutely analogous to retroviruses, from the RNA to DNA, hexanucleotides, and that's encoded by a gene called TERT, T-E-R-T, the telomerase gene. And these with other structural proteins that stabilize the molecular machinery. One of them, in fact, is DKC, the gene that Dokal identified in some X-linked cases of DYSKERATOSIS congenita, form this molecular machine to repair telomeres, so an active process. And what happens when a cell undergoes mitosis is that despite the presence of the telomere and despite the repair process, there is an inevitable loss of DNA at the termini. there is an inevitable loss, even with the repair. When that loss over the course of many, many cell cycles, as for example occurs with the fibroblast in cell culture leading to the Hayflick phenomena, when that loss of telomeres becomes critical and the chromosome is no longer coded by the telomere and its associated proteins, the cell is signaled to go to sleep or kill itself. I hope that's not too, think about the audience, are we sleeping? So the cell is going to no longer proliferate. Not a problem in most organs because there are plenty of cells, and that cell that's reached the end of its mitotic life simply drops out of the picture and other cells will compensate for it. But think about that in the structure of an organ, because I'll return to it at the end. Under specific circumstances, as for example with suppression of the p53 DNA damage response and the cell's survival, the cell becomes genomically unstable. The chromosomes will undergo non-reciprocal translocations. Aneuploid events will occur. The cell will now have major problems with chromosome stability. That, if there is no DNA damage response, can lead to pre-malignant lesions and frank cancer. As I mentioned before, our chromosomes are suffering telomere attrition under normal circumstances. This is our own data with Peter Lansdorp showing you telomere content. We say it's length, but it's really content that we're measuring. You see that this is falling over the course of natural lifespans up to the age of about 100. Most of the loss occurring early on. You tell your adolescent children that they're in fact aging faster than you are. There's a sort of stability in middle life and then a drop off, whatever age I am, 1 year older. And the point, this is the paper that we published looking for telomerase gene mutations. In patients who have defects in telomerase or in telomere repair, this process is greatly accelerated. So in contrast to gene phenotype relationships that are all too typical, where you have some gene that you don't really know the name, don't really understand what it does, it's got some yeast function, we think there might be a homolog or an analog in the adult. With telomerase diseases, we understand the physiology relatively well and even have a commercial or laboratory-based blood test that tells us the result of the gene's dysfunction, and that is telomere content in peripheral blood leukocytes. So this is just an example, but now it's widely used, of very, very short telomeres detected in this instance by flow fish, but there are other methods as well, that allow you to determine the telomere content of just a peripheral blood leukocyte sample, and almost invariably, that's extremely low, below the first percentile for whatever the age-matched control is. So when telomere disease is suspected, and I'm going to suggest to you how you actually will be able to make that diagnosis in the future, or even in patients that you've already seen in your own clinics, across the spectrum of medicine, you can really make the diagnosis with a relatively easy blood test. Now I want to give you some clinical vignettes after this molecular biology of the way telomere disease can present in the clinic. Now, there's nothing new about telomere disease. As I'll show you, we can now recognize in retrospect patients who very typically have the disease. You can make this diagnosis when you walk into the patient's room, and it also provides an explanation for some of the old stories, the old shibboleths in bone marrow failure, and I think in other diseases. Now, this is a very dramatic story of an American Army officer who was actually airlifted out of Afghanistan and came to us, has no hematologic problem on presentation. He actually presents with tongue cancer. And in children with DYSKERATOSIS congenita, the rate of tongue cancer is about 1,000 times higher than it is in the normal population. He has not only tongue cancer, but he almost immediately metastasizes to his lungs, which is what are shown here, undergoes a round of chemotherapy, and his blood counts, which had been normal, bottom out, and he develops long-term pancytopenia. Now, I wish I could say I was the one who made the diagnosis, but it was a very astute Army physician who looked at this patient's nails and looked at his skin and realized that he had classic manifestations of DYSKERATOSIS congenita. And in fact, we demonstrated that he had a DKC1 gene mutation, explaining his short telomeres and his clinical picture. So this is late presentation in adulthood of a congenital syndrome. This is the much more typical story, and this is how you can make the diagnosis walking into the room. So here's a man who presents with chronic, progressive, but not tremendously severe thrombocytopenia, platelet counts falling over the course of a decade from the 100, 90, 80,000 range to 30 or 40. He's completely asymptomatic. He has big red cells, but has no anemia. He does not have a positive family history. And if family history is not required, in patients to make the diagnosis. But his hair, this is his own iPhone photo that he took after we demonstrated his telomeres were extremely short, and that he has a mutation in the TERT gene. He had early graying of his hair in his early 20s. And later we found that he also had cryptic cirrhosis, as I'll show you one of the other manifestations of telomere disease. So this picture of thrombocytopenia, anemia, or macrocytosis without anemia, often slowly progressive, usually called moderate aplastic anemia or even myelodysplasia, is very characteristic of the telomere diseases in a hematology clinic. Early graying is not invariable, and obviously there are other reasons for early graying. I actually queried Tim Lay about his gray hair, so there are other reasons that your hair can turn gray early, but early graying, when it's seen in this context, is a very good signal. Now, there are old stories, the older hematologists in the room will remember these case reports in small series that blamed hair dye for aplastic anemia. And if you smell hair dye, my wife now dyes her hair, if you smell it, that should cause aplastic anemia. It's really foul. So it seemed reasonable. But the story really is a reversal of causation. And it's important to ask not only women but men whether they dye their hair, because if your hair turns gray and you're 19 years old, you may be tempted, in fact, to cover that with hair dye. So I think the old story of hair dye is one of patients who had telomere diseases and were, in fact, confronting the particular cosmetic manifestation of that and then later developed marrow failure. This is a very large family. I'm not actually showing you the entire family. Half of them had gone back to their farm work. They're a family that lived near Washington in rural Maryland, dairy farmers, Mennonites, as you can tell from their costumes, who have a mutation in telomere repair that in this very large family has allowed us to see many of the clinical manifestations. It's a good way of introducing the non-hematologic diseases to an audience because they're all present in this huge pedigree. Those of you that had the opportunity to study Mennonites or the Amish know that not only do the families stay together for their religious reasons, but they also keep fabulous personal records. So they have a genealogy book that they self-publish that goes back several centuries. They also kind of have their philosophy based on, we think of them as being very old-fashioned, but their philosophy is actually based kind of on Spinoza's principles. Spinoza ran off to be with the Mennonites when he was excommunicated from the Jewish community in Amsterdam. So it's a very holistic, pantheistic view of nature, and so they're very responsive to being studied. In fact, by inquisitive doctors. And when I gave a lecture talk conversation with them about the potential for telomere disease, a farmer in the back raised his hand and said, "Is this related to our mitochondrial problem?" So they're actually surprisingly sophisticated despite their old way of dressing. So this is the family. Everybody who is in green has a mutation, a novel mutation in telomerase, and the TERT gene is shown. Everyone in white is normal. So this is a huge pedigree. So who's sick in this family? The proband was a young dairy farmer who presented with very much the story I've just told you about, that pipe fitter from the Midwest, who had progressive anemia and thrombocytopenia, came to us. He's got a big shock of gray hair. It was easy to make the diagnosis. Again, as I walked into the patient's room, he had had no response to the usual therapies, and we actually ultimately took him to transplant. My colleague Richard Childs did a very uncomplicated transplant, a very important demonstration of the value of genetic testing, even in a transplant clinic. You can see this is a big family. Here's our patient. He had several HLA-matched sisters as potential donors, one of whom, shown here, was hematologically completely normal. And if she had been used as a donor, he would have died. We know from the examples of others that when an unrecognized telomerase mutated patient is a donor, they have insufficient hematopoietic cells. Patients either don't engraft or have early graft failure. So he chose instead one of his genetically normal HLA-matched sisters. He's essentially had an uncomplicated transplant. And this is the point that I want to make, is that these genes provide a risk factor. They do affect hematopoiesis almost invariably in patients or individuals who carry them. They may not be patients. This is from one of our families showing that the proband has an extremely hypocellular bone marrow. So does a genetically affected sister and a niece. Here's an unaffected control in the same family. But the patients or individuals who have these mutations can have completely normal counts or very minimal macrocytosis, big red cells, or very minimal anemia. However, if you look at hematopoiesis in the laboratory, they not only have a deficient bone marrow appearance, but have low numbers of CD34 cells, colony formation, very high concentrations in their blood of the growth factors that drive hematopoiesis. Now, returning to this family, I've told you a bit about the blood disease in this patient. I'm going to return to that in a moment. There are also 4 instances of severe liver disease. Ultimately, they all proved fatal, all occurring in women in the family, including a case of fulminant hepatitis in a young woman at the age of 19. She was rescued by a liver transplant. So you see them that they're all marked here with these stars, and they all track with the mutation. So this was a suggestion that a second organ, not just the bone marrow, but the liver, was also affected in patients with telomeropathy. This is a remarkable family of that we wrote up back in the 1990s when we recognized this link between liver disease and liver disease and telomerase mutations. I recalled these 2 patients, these 2 families, that we'd written up in a journal. I'm not even sure if it still exists. It's not one of my most highly cited papers, but we saw this link between cirrhosis and aplastic anemia, just a clinical link. And it's a good test of aging, if not necessarily telomere length, but I was actually able to remember the name of the family. Of course, you try to call 15 to 20 years later, the phone doesn't answer, the doctors died, so I assumed that they were lost until I got a phone call from a colleague in our transfusion medicine department who said that there was a young woman there who wanted to donate her bone marrow for some other person to benefit, and that her brother and her father had been seen by me 15 or 20 years ago. So, of course, it was the same family member. She came to see us, and I learned I'd only seen the family on a couple of occasions. I'd learned the sad outcome in this particular pedigree. So the patient that I had originally seen was this young man here who had just very mild thrombocytopenia as an 11 or 12-year-old. He went on to develop, not under our observation, very severe aplastic anemia, went to an unrelated transplant and died shortly after. It's a very sad story. His father had also suffered aplastic anemia and severe cirrhosis. That's a terrible combination. You have esophageal varices and are bleeding and have no platelets. That's not going to be a good outcome, and he had died as well. And you can see that there are other organs. I'm going to come back to the lung in a moment, but also cirrhosis and other family members. And now we're presented with this young woman. She's got her 2 young kids. Everybody looks healthy. And we measured her telomeres, and they were indeed short. But she's going to be fine. So why is she fine? Because her gene is normal. And why does she have short telomeres? So if you're following the conversation, you inherit your telomeres, not only your genes, but you inherit your telomeres from your father's sperm and your mother's egg. And her father's sperm had extremely short telomeres that could not be easily regenerated in his gonads. So she had short telomeres, but she could repair them. And from mouse as well as other human observations in families, as long as the repair capacity is intact, the organs and the cells are going to be fine. And you can show this, for example, in mouse breeding studies. So she will not manifest any disease, and her children do not need to be tested. So the relationship between cirrhosis and the telomerase gene mutations has been demonstrated not in patients with hematologic disease, but just in patients with cirrhosis. And both our group and Lenhard Rudolph's group in Germany had very similar papers appear in the hepatology literature, showing that somewhere between 5 and 8% of patients with cirrhosis, whether it's hepatitis or diabetic steatosis or alcohol as a factor in the development of cirrhosis, that there are underlying mutations in mainly the telomerase gene, the TERT gene itself. And we've just recently begun to model this in mouse experiments, which are really quite striking. I'm only showing you one example of these unpublished data from our Brazilian colleagues working in our laboratory, especially my friend Rodrigo Collado, that the TERT-deficient mice, when they're fed a high-fat diet, develop, this looks a little bit like a bone marrow, develop this fatty appearance. We don't see that occur in animals that have the RNA gene defect. And this actually parallels what we see in the clinic, which is that TERT, but not TERT, mutations are related to the development of liver disease. And there are also quite striking metabolic profiles that change early in these livers when the animals are stressed, as in this instance, with a high-fat diet. So I think we're going to be able to work out what goes on in the liver using these animal models as well as good clinical phenotyping. Now, I mentioned that there's a third organ in that family, pulmonary fibrosis. This is actually a patient who had undergone, who had been treated for aplastic anemia 20 years. This is part of our AMENNONITE family. Treated for 20 years with a drug that I'll come back to at the very end, had done well with her bone marrow failure. came to an NIH meeting about DYSKERATOSIS congenita for patients, in which something like this picture was shown, indicating that lung is also one of the organs involved in the telomeropathies, turned to her husband, she was a very stoic woman, and said, you know, I've really been feeling short of breath for the last couple of months. This is the CT scan that we did, showing this classic honeycomb appearance of pulmonary fibrosis, and she actually was dead 1 month later of pulmonary insufficiency. Incidentally, she was found to have occult cirrhosis. She actually suffered in all 3 of the affected organs disease. This is work from others, from Jerry Shea's group and also from Carol Greider at Hopkins, showing that again on the order of 5 or 10% of patients with hereditary, with inherited pulmonary fibrosis have the same sorts of mutations that we see in patients with aplastic anemia and in cirrhosis, in this case more often TERP, the RNA, rather than the telomerase gene itself. So these 3 organs are affected. This is the important slide to remember. There's telomere erosion that's accelerated as a result of telomerase deficiency. There are environmental factors for the marrow, for the liver, I've mentioned alcohol and hepatitis, that increase the regenerative stress on the organ, which is now not met adequately because of these underlying genetic mutations, leading to stem cell loss and in the lung and in the liver fibrosis, and therefore organ failure. The penetrance and the phenotype in families and even in individuals is extremely variable, which makes genetic counseling difficult, but also makes diagnosis often problematic. So there can be very subtle findings, for example, in the liver or in the lung, which are not progressive and association with aplastic anemia, or you can have severe cirrhosis with very mild changes in blood counts. This is a way of looking at it in which Rodrigo and I plotted for this review paper, genetic penetrance and environment. It also allows us to separate out, and I think this is very important to do, to separate out the very highly penetrant DYSKERATOSIS congenita pediatric syndrome, which very rarely will manifest in an adult, from the telomerase gene mutations TERT and TERP, which characterize risk factors for these 3 organs. And note also that the skin and mucosal membranes are rarely involved in adults, making the diagnosis a little bit more difficult. We've got a couple of features in terms of where we're actually going with the study of telomerase mutations in the clinic. This is from one of my junior colleagues, Danielle Townsley, presented at ASH. This is a huge study of now almost 200 patients with telomeropathies in our clinic. Of course, this is a very skewed presentation. We're a hematology clinic. We don't see patients with liver or lung disease primarily. But even from the point of view of the bone marrow, there's a very wide spectrum, from moderate to severe aplastic anemia, sometimes associated with hepatitis, mild thrombocytopenia or macrocytosis, pure red cell aplasia in a couple of instances, and some other syndromes that seem to be related to this genetic defect. Second point is that when we see short telomeres, we can't always find a mutation. And telomerase regulation is extremely complex, involving many different genetic pathways. This is just one cute example from our clinic of where the molecular biology can get very complicated. So here's a family that has absolutely typical telomere disease. There's scattered through this family pulmonary fibrosis, aplastic anemia. This is our proband. Very, very short telomeres. No question about the diagnosis, but the TERT and TERP genome sequence, gene sequencing is normal. So we had a very bright young woman in our laboratory from Holland named Anna Albers, who looked just a few hundred base pairs upstream in the regulatory region of the promoter and found actually what turns out to be the first mutation in the CAAT box, CCAAT, regulatory element in the promoter of the TERP gene, which disrupted the function of the TERP gene. So regulatory elements also can be involved. This was relatively easy, but now looking way upstream in cancer or suppressor elements, looking at other genes like MYC, for example, that affect telomerase, that's going to be really quite complicated and require very sophisticated genomics. But in general, we make the assumption that when we see a family like this with the corollary short telomeres, there's a mutation somewhere. We just don't know where it is. Mutations are off. This is also unpublished. This is unpublished work. This is also, again, an extraordinary story. Patient, not of ours, but of the Cancer Institute, who has another genetic mutation leading to myelodysplasia, not one for me to discuss today, who goes to transplant and has a very complicated course with much delayed engraftment of umbilical cord blood and ultimately leukemia developing in the donor cord cells. And what's striking in this patient is that the umbilical cord blood that went into this patient was extremely deficient in telomere content. Now, the child, as far as we can tell, that's developed from that umbilical cord blood is fine. We don't understand whether this is a genetic or more likely a proparosome lesion, the way the umbilical cord was actually dealt with. But these telomeres are almost half, or just about half the length of what should be very long telomeres in the umbilical cord blood. And there's very little in the way of mutations at the time of leukemia. So it appears that in this instance, as I'm going to suggest in a moment, this accelerated telomere attrition that occurs as a result of short telomeres to begin with, and then the stress of regenerating the bone marrow with this umbilical cord blood contributed or caused the leukemia. And then finally, in terms of future directions, we and now others have developed inducible pluripotent stem cells with skin from patients with TERT and TERT mutations. These likely will prove very valuable. This is work that was just published in JCI. Will prove very valuable in looking at some of the control mechanisms, including the physiologic control mechanisms. We see that there's very deficient maintenance of telomere length. Normally, inducible pluripotent stem cells generate telomere elongation because they've got active telomerase, but when there's haploinsufficiency, you see that there's not telomere elongation. And in fact, these cells appear to be susceptible to chromosome rearrangements, and there are interesting phenomena like, for example, low oxygen tension, less in the way of reactive oxygen species, less damage to the DNA overall. Also helps in terms of maintaining telomeres. There's actually some evidence of telomere elongation when the amount of reactive oxygen species is reduced in hypoxic conditions. Okay, so I want to return in the last few minutes to this cancer link that I've been tantalizing you with through the course of this lecture. So I told you about the patient, but he actually had a very important clinical vignette within his family, which was the course of his father. So his father, a man in his 30s, had developed what looked like acute leukemia emerging out of what we would now call myelodysplastic syndrome. He was actually treated at a hospital not very far from Washington by some prominent American hematologists who all remember him. Again, this very characteristic family, big family, all dressed in Mennonite costume, and the really awful course that this man had, which is that he got a single course of chemotherapy for his AML. His counts failed, became cytopenic, and he died of infection. And that's quite unusual in a young person with leukemia. Generally, we can get them through a single round of induction chemotherapy. The problems occur in the next 6 or 12 months. So we identified in archival tissue that, of course, he has the same mutation as his son, and that's where his son, in fact, inherited the TERT mutation. So Rodrigo, in the laboratory, collaborated with groups at MD Anderson, his native Brazil, São Paulo University, and Vancouver, to look at a very large number of patients with de novo leukemias, and no telomere suspicion here at all, and was able to show, and I'm not going to go through the slide in great detail, but in these many hundreds of leukemic cells that were examined, germline mutations, again on the order of 5 to 10% underlying leukemia in these cases. Now, of course, we think about telomerase in cancer in general as being upregulated. That's the way cancer cells maintain their mortality. They have to keep repairing the chromosomes. But the thought here, and there's abundant animal and laboratory data, is that in the early events in oncogenesis, failure to maintain telomeres, in this instance presumably due to germline defects, leads to this chromosome instability. And in fact, almost all these patients have chromosome abnormalities, which only occur in general in about 50% of patients with AML. So TERT mutations appear to underlie a subset of patients with acute leukemia. So we have this problem in aplastic anemia that I mentioned to you of evolution from what appears to be an immune or inflammatory disease with bone marrow failure to, in about 15% of cases, myelodysplasia or leukemia, almost invariably associated with very characteristic chromosome abnormalities, especially monocyte 7. This is the problem of clonal evolution. So Phil Scheinberg, who despite his name is actually also Brazilian, looked at several hundred of our patients who do not have genetic mutations. These are just patients that we've screened. They don't have telomerase defects, but they've gone through immunosuppressive therapy protocols at NIH. And he asked the question of whether telomere length, independent of the genetics, but just having shorter telomeres, even if they're in the normal range or just below the normal range, whether that had any impact on how patients did long term. And in this work that was published a few years ago, what he found was that the telomere length was irrelevant for the response to immunosuppressive therapy. And just as an aside, as a parenthesis, patients can have TERT and TERT mutations and respond to immunosuppressive therapy. It's really quite convincing evidence that the immune system is an overlay on this underlying genetic deficit. But this is not an underlying genetic problem. These are just patients with shorter telomeres. So no difference in response to immunosuppression. They were more likely to relapse, but what was particularly striking is that they had a much higher rate of evolution to monosomy 7 or to MDS, almost 5 or 6-fold higher as shown here. And if we go back into our freezers and look at their bone marrows months or years before they develop MDS or AML, we can not only detect the short telomeres. You see that these little double-headed worms are lacking eyeballs. That's evidence of no telomeres at the end of the chromosomes, in contrast to another patient's bone marrow where there are always 4 fluorescent signals per chromosome. But if we look at the culture of these cells, for example, using spectrokaryotyping, we can detect the sort of classic non-reciprocal translocations or chromosome loss years prior to the development of their monosomy 7 or MDS. Now, there is a huge world out there of biologists who study cancer who believe that the chromosome is really the basis for cancer and the mutations follow. And of course, you've heard from others recently that there's also a very big world within hematology that believes the cancers are just a result of one mutation after another. And one of the problems in these studies, of course, is getting the right human system or the human model to look at, and also being able to look early enough. So it's one thing if you take out a colon cancer and compare it to the adjacent normal colon epithelium and find mutations. You can certainly say there's a difference between the cancer and the normal adjacent tissue. But are those mutations the reason that the patient developed cancer, or is that part of the process of evolving within a cancerous lesion the most effective proliferating and surviving cells? So obviously, as I've indicated to you, we have a tremendous opportunity in bone marrow failure to look at these very earliest events in oncogenesis. I'm just going to show you a little bit of the most current work. So Bogdan Dimitri in our lab has taken 13 patients who've developed classic monosomy 7. It's chromosome lesion, terrible prognosis. Really, we don't understand exactly why it develops. It presumably provides some tremendous advantage to those cells that lose chromosome 7 without killing them. And the patients are pretty well matched to a group of about 2 to 3 times the number of patients with stable aplastic anemia, except that they don't survive as well. And we have serial samples that we can go back on and look and compare directly what's happening to the patient's chromosomes compared to what's happening to the patient's genes. So when we look at the chromosomes and we look at telomere content, you see that the patients who have clonal evolution have much shorter telomeres than the patients with stable aplastic anemia. Again, there are no mutations in TERT or TERK involved here. This is just typical aplastic anemia. And if we look serially at those samples of blood that we've collected over the course of many years, you see that there's a very striking difference in telomere loss in patients who go to clonal evolution than those patients who have stable aplastic anemia. This is looking at the mean telomere content of all leukocytes. So we can actually look at individual chromosomes. This is looking at the X and Y chromosomes, and you can see that these fragments, this is a method called STELA, or single telomere length analysis. This is a presentation, and you see in this patient with clonal evolution the acquisition of these short telomere fragments occurring quite early in the course, a couple of years before the patient goes on to develop monosomy 7, and that's quite typical of patients in general. Now, this is again measured by STELA compared to patients who have stable aplastic anemia. Now, in the same group of patients, we can ask whether they have mutations in the MDS AML candidate genes. And they're probably only on the order of 20 or 30 genes that explain most of the recurrently mutated genes that are factors in most MDS and AML. So we've looked at those specific genes, and this has actually been done in 2 separate sequencing facilities. And what I want to emphasize here is that the picture is not absolute, but it certainly favors chromosomes. So as shown, and I'm going to describe this in greater detail for those of you who are at my different lecture tomorrow, there are mutations that occur in some patients in these classic genes, as for example in the first 2 patients, and they're big clones. So we can make an argument that these mutated hematopoietic cells are in fact related to the development of MDS and AML. But shown in green are very small clones that are hard to impute when a patient has 100% monosomy 7 cells and 5 to 10% blast. Probably most of the bone marrow is now involved with the mutant, with the abnormal aberrant clone. These clones only constitute 3 or 4%. of the bone marrow. So the cells, the patients shown with the small clones shown in green, and also those patients in whom we're not able to find any mutations, presumably do not have a mutation-driven development of monosomy 7. So this is just a summary of a complementary study. Again, I'll describe this in greater detail tomorrow, looking at hundreds of patients by genomics with aplastic anemia, and just summarizing what I'm going to talk about at greater length tomorrow, which is that while mutations occur in these patients, the clonal architecture of the bone marrow is quite complicated and doesn't follow the sort of standard picture that you see with MDS or AML. Mutation, clone size, another mutation, clone gets bigger, third mutation, frank MDS or AML. In fact, in the marrow failure setting, these clones are small, they're not present in most patients, and they can come and go, and some of the clones almost always disappear for reasons that are really peculiar and don't seem to have any relationship to any of the clinical correlates. Does the patient develop MDS or AML? Do they relapse? Do they respond to immunosuppressive therapy? Not related to the simple presence of these clones. This is an example, very striking example of what I'm talking about. This is actually whole exome sequencing in one patient we followed for over 10 years, where I think you do see both the irrelevance of the clones and also their relevance. So this is a patient who's been followed now for 10 years, had a great response to immunosuppression, but required cyclosporin. He's now a man in his 80s. And you see that, for example, these PET2 and B-core clones are present in small numbers at the onset of the disease, and they disappear. And that's quite typical of B-core mutations. We have an ASXL1 clone that is at a pretty good level for most of the time that this patient is sick, while he goes through treatment, responds to treatment, has good blood counts, managed as an outpatient for 10 years. Hard to know what the relevance of that is. And then the SETBP1 clone, which does appear to be the driver for the development of MDS in this patient whose marrow done 4 or 5 days ago now shows 5% blast in his myelodysplastic. So mutations have a role, but they don't seem to be predominant in most patients. And I'm going to talk about tomorrow also our clinical efforts to actually stimulate hematopoietic stem cells with a magic little pill called L-Trombopag, and we do see problems with clonal evolution under some circumstances. So patients who are treated with a stem cell or early progenitor stimulator do develop monosomy 7 if they have long-standing disease. And we see that there's a difference between those patients with long-standing disease and patients who are treated with this magic stem cell progenitor pill early in their course. And we think that that's based on telomere length. So patients, as you would expect, with chronic refractory aplastic anemia do have shorter telomeres, shown here in yellow, whereas patients with new disease have more normal telomeres. And those patients who evolve also tend to be the ones with the shortest telomeres. Now, again, I want to stress that I don't think this means that the patients have mutations that we haven't recognized. I think that what we're seeing with these short telomeres is evidence of the number of hematopoietic stem cells that are lost, as well as the stress of regeneration that occurs in these bones. All right, so there is this chromosome theory of cancer. I'm not going to have time to go through this, but, of course, somebody discovered everything that you thought you could take credit for. 100 years ago, this poor guy published this fantastic volume right at the onset of the First World War. Not a good time to come out with a new theory. Really sort of lost. There's tons of data, I'm not going to go through this, we don't have time, that come from both animal and mouse and human studies implicating telomere loss as a general mechanism for cancer development. I've just summarized that here in this diagram. I've told you about the constitutional problems with TERT and TERK and other gene mutations and the proclivity in that setting for lesions, as for example AML. We have the setting of physiologic aging, and after all, age is the major correlate with cancer, with very rare exception, just getting older. And of course, we have a such situation that I'm particularly interested in, which is what we create ourselves as oncologists when we stress patients and treat them with chemotherapy or radiation therapy, creating bone marrow regeneration, telomere erosion, and this chromosome instability. That's almost always associated with complex cytogenetics. It's going to be problematic separating out what are two concurrent events. Every time a cell goes through mitosis, it loses telomeres, but it also has an opportunity to have a mutation. You need a mitosis for that to occur. So it's going to take some ingenuity to figure out which is really the driver. So this is the general way of thinking about cancer and telomere loss. This is not work just from my laboratory, but from many laboratories. And I think it's particularly this middle group of patients who have immune inflammatory diseases, and who have a risk for cancer that may be best explained by telomere attrition. I'm giving you just some examples here. You can think of others. Peptic ulcer disease, esophageal erosion and esophageal cancer, ulcerative colitis, bowel cancer, and so on. So I want to end on a more positive note. When I hear the genomic story, it's really quite depressing. You get older, you acquire those mutations, bad luck, get 3 mutations in one gene, that's it. But we may actually be able to do something about telomeres. So this is again from our Mennonite family. This is a woman who's holding her homemade diagram of her hematocrit when she was treated 15 years ago with male hormones, with an androgen. Very popular in hematology for the last 50 or 60 years, and they work in a subset of patients with bone marrow failure. This is one of the signal papers published back in the 1960s. And we've never understood why they work. They certainly increase erythropoietin, but that's probably not the main mechanism. The white cells and platelets increasing. So Rodrigo in our laboratory showed that in tissue culture, if you add male hormones, but also female sex hormones, to hematopoietic cells, you actually directly upregulate the telomerase gene, the TERT gene. That's the critical regulator of telomere repair. And the explanation for that is a couple of nice estrogen response elements, aromatization of the androgen in the cell to an estrogen estradiol and binding to this ERE in the promoter of TERT. And we're looking at the clinic in a more prospective and a more systematic way at the influence of a sex hormone on hematopoiesis, on the lung also, but mainly on telomeres. So this is a study that Bogdan is conducting in our laboratory, looking at 2 years of danazol, a nice synthetic male hormone, and these are the results. So hematologically, if you take patients who have TERT or TERT mutations with very short telomeres, the response rate to male hormones per protocol is quite high. Almost everyone who can tolerate danazol will respond. But what's most striking biologically is that it appears that we've, and I think this is the first example in humans, we've reversed telomere loss occurring in these patients who have, we think, accelerated telomere attrition and have either stabilized or actually elongated telomeres in this setting. Now, nobody wants to treat everybody with danazol. There are obviously other ways to upregulate telomerase that either can be investigated now or can be determined. But this really suggests that we can do something about telomere loss in specific settings, as, for example, post-chemotherapy when patients are at risk for secondary MDS and AML. That's everything I wanted to tell you. I really thank you for your patience. I know I've thrown a lot of slides at you. I hope some of it at least is clear, and I thank all the people who actually have done the work in my laboratory over the course of the last 6 or 7 years and show you your clinical center. It's yours in the sense that the taxpayers have supported this institution and supported me for multiple decades. And thank you again, John, for the invitation to be the Clem Finch visiting professor. All right, that's probably a separate lecture. This issue of telomeres and aging is a really problematic one. Now, if I had to support myself on telomeres, I'd want it to be related to aging, and I want to have all cancers be a result of short telomeres. I think the cancer issues I've shown you is going to be complicated because of this problem of mutations and telomere loss occurring simultaneously in cells. Aging is even a bigger problem. And there's no question that telomere loss accompanies aging. The question is whether aging is caused by telomere loss. And the answer to your question is that most people, we don't know that. But I don't think it's fair to characterize the telomeropathies as a premature aging syndrome. The gray hair makes you think that, but hepatic cirrhosis, even marrow failure, and pulmonary fibrosis are not consequences of aging. You don't see those on autopsies. And there is no evidence that, for example, the gut, and we think that that's a highly replicative organ, that suffers at all in patients with telomeropathy. So no, I don't think that I've got nice long telomeres, so I'd like to believe that I'll live forever, but I really don't think that the data are very convincing that this is really a cause of aging.
What's known about fibrosis? Do patients have chronic fibrosis or
Yeah.
scleroderma?
Yeah, so the myelofibrosis, the answer is no. We've looked and others have looked for mutations, and you'd expect that, right? I mean, you've got fibrosis in these two other organs. So why the default pathway is fibrosis in two organs? And by the way, in the lung, we have no idea which is the cell that's critical. I mean, my lung colleagues tell me there's 73 different types of cells in the lung. I don't know which one short telomeres has an effect. And so we don't understand what the trigger is. You know, is that cytokine mediated? Is it cell-cell? It's really not understood. Myelofibrosis is not related. Scleroderma is a little bit more problematic because there is pulmonary fibrosis that can occur, and there are old reports of short telomeres detected by Southern in patients with scleroderma. We've tried, if you've got scleroderma patients, we'd love to look, but there's not been a recent study of telomere length in that disease.
Neal, so is telomere length lost general across all chromosomes, or is there some chromosome-specific loss? And does that correlate in any form with the
Yeah, so the answer is that telomere biology is complicated, and the answer in humans is that that's not known. In the mouse, every telomere appears to have its own genetic regulation of telomere loss, and so telomeres are not equivalent on all chromosomes in terms of their length or their repair. The telomerase machinery also is really smart, and it goes to those chromosomes that have the shortest telomeres. So as they erode on one chromosome or another, that's where telomerase is preferentially active. So that is really not understood. You know, is there loss on specific chromosomes that leads to instability? It'd be nice to think, for example, that maybe chromosome 7 loses telomere faster, and that's why we see loss, but it's really not known.
Thank you very much, Dr. Young.
Thank you. Good, thank you.